You would imagine that with mankind's awareness on science and technology, we are sending rockets into space. You would imagine that we know why living beings need oxygen. I will tell you that we don't. Of course, we have a solution to it. So the topic of the presentation today is, why do we use oxygen and how does cyanide kill so quickly? I have put this title so that I can catch your attention at the outset. All right? So, and this question I shall aim to answer with Murburn model of aerobic respiration. So, let me just go on. I will come back to the topic. Next slide, please. My background is in enzymology. So you know what are enzymes. Majority of you are from bioscience background, right? So you know what are enzymes. While I was doing my master's, I did proteases, PhD on lipases, postdoctoral on epoxide hydrolases in various parts of the world. And then, please, okay, thank you so much. So, now, if you study enzymes, reactions are based on collisions. That's a fundamental theory, collision theory, and then there is a transition state theory, right? You have transition state complexes formed. That's one of the fundamentals. And particularly when you come to enzymes, you have lock and key concepts, Fisher's lock and key concept, or then the induced, Koshland's induced Swift theory, how enzymes recognize their substrates, right? These are the fundamentals. So, all that changed when I went to Urbana-Champaign. This is Lowell P. Hager, my mentor. Like what Danny sees me as, I see Lowell, my, guiding light. When I was working with him, he gave me absolute freedom. He allowed me to challenge him, to prove him wrong, all that. And that's very essential for a scientist. If you cannot question what you get to see from textbooks, what you hear, you're not a scientist. And this man, in his lab, getting funded from his project on the enzyme that he discovered, I challenged his ideas, 50 years standing ideas, and I proved him wrong, and he graciously accepted it. And that's what a scientific temperament is. So, and you know what? When I go to my classes, this is what I say. If you ask me a question I don't know, you get full marks in assignment. If you find something wrong in what I say, you get marks in assignment. That's how science is essentially a negationary pursuit. You can say what is wrong, and that's what the students should be always inculcated. You should find faults. Finding faults is what science is about. So, working in his lab, one night I found that azide, heme ligand, it's a little technical term, it is supposed to bind the enzyme and inactivate it. I found that at a particular low concentration of azide, it's something like cyanide, okay? The enzyme activity was enhanced 10, 20 folds. I couldn't believe it. And I reported it to my PI. He said, nah, it could be an artifact, right? And then I didn't let go. I did it again. I believed my eyes. I could repeat it. So this is one thing I want to tell you. One simple thing that you will find in your lab when you do something, that is what is going to make your life, if you believe in yourself and if you believe in your finding. This simple thing that I did one night in his lab, that's what I am going to talk to you about. You are the first speakers on an invited thing that is going to get the information directly from the discoverer of the fundamental phenomena why oxygen is needed for life forms. So, next slide. You will see this on the Wikipedia page of Murburn concept. What is Murburn concept? Murburn concept is a simple idea, a group of ideas that is poised to explain a wide bevy of metabolic and physiological events and routines in life. Okay, so Murburn concept, conventional enzyme, this is a classical enzyme where you have an active site to which a substrate comes and binds. This is it, and it gives products. This is a simple enzyme reaction, right? Right? Now, what happens in Murburn concept is you have an enzyme, it might or might not bind to the enzyme, but there is a diffusible species that is generated, and this diffusible species interacts with the substrate to give products. That's all. So, it's a very simple idea. That is, an enzyme produces a diffusible species that reacts with the substrate. It's a new concept in enzymology per se. Very simple idea, but the ramifications are profound. I will go into the next slide. Now, the choice of the word Murburn, of course, my name is Murali, so I put MUR, but I found a reason for it. All our living beings are made of molecule ions, right? Molecules and ions. That's what you are essentially made of. So molecules, unbound ions, and then there is something called radicals. So molecule, unbound ion, radical. That is Mur. And then there is another thing, mured burning. What does mured mean? Mured means closed burning. So burning is something that involves oxygen. You would have seen in day-to-day life. Burning is something that involves oxygen. What happens in a vehicle? Suppose in a scooter there is an engine, right? You put oil in it and then the oil is burnt using oxygen. And then you can do some useful work of transporting a person from point A to point B. That's what a working machine does. Your body is exactly similar. You eat. And that eating gives you fuel. That food, glycolysis, Krebs cycle, all that you have studied, ultimately it has to make ATP. ATP and heat energy. That's what this process is about. This Murburn is a catalytic scheme that involves enzymes with heme flavin cofactors and then substrates, wide variety of reactions it can do. The main involvement is of oxygen and this ROS is, reactive oxygen species, very trendy word. So there you have molecules, ions and radicals that are produced, and there is an interactive equilibrium between that, and then this can do a wide variety of reactions. Usually standard enzymes will do only one reaction, reacting with one substrate, right? You might have studied, right? So this is very different. The reactions can be diverse reactions, diverse substrates. So this is it. Now I'm going deeper. First slide, I showed you the simplest explanation, Murburn concept. Then it can have intricacies. It can have even more intricacies. I'm not going to go into that because not many of you are specialists, but you must see, once the reaction occurs outside the active site, these ions can interact, these radicals can interact, and several molecules can interact. Now you can understand the intricacies. Now, conventional enzymology cannot explain certain things. This interactive paradigm can give you a wide variety of outcomes. It is a fast process because radicals are involved, right? Radicals give fast, and then normally when you write an equation, two molecules of this react with one molecule of this to give two molecules of this and one molecule of that. You can write. Here you can get non-integral stoichiometry. What is non-integral stoichiometry? 0.5, 0.75, 1.3, you can get like that. Why? Because this is a statistical and stochastical paradigm. Statistical means stochastical means probability, more or less the same thing. So you get atypical kinetics, you get different and unusual dose responses. So I'm just showing you some intricacies, I will not go into that. The more fundamental aspect of it is on a philosophical plane. It has, this Murburn concept has order at one point and it has chaos also. Because diffusible radicals are involved, it can do a wide variety of reactions, but then it also gives order. So both order is involved and chaos is also involved. And then modern day science deems refusible reactive oxygen species as the harbinger of practically all pathophysiological states in life. So what I vouch for is that these are the quintessential elixirs of sustaining life. So now, why is it not changed? Okay. Now, you know the reactions need selectivity, specificity and stoichiometry. What is selectivity? If there are different shape molecules like this, out of that you want a particular molecule to be reacted. Life requires that, right? You don't want all reactions to happen. You want selective. So, if you take that particular molecule also, it can be attacked here, there, so many places. You can get wide variety of products. So, I have shown that this kind of selectivity, specificity, this kind of different stoichiometry that you get are inherent in certain reaction media, that is by virtue of thermodynamic features, delta G of formation, thermodynamic pull, there are proton spin locks, like that. There are different reasons. You might have seen the Michaelis-Menten curve, but certain times what you do get is, it might not be like that. You might get crazier curves like this, maverick curves. These are explained by Morborn concept. And more importantly, dose response. Sometimes you get 1 gram you eat of a drug, it might not give you a therapeutic effect. 1 milligram you take, you might get an effect. How is that? Morborn concept is the first explanation for that. Why? Because radicals are stabilized at lower concentration. At lower concentration, you might get an effect. So, things like that. I'm just showing where Morborn concept has been applied before coming to the main topic. Okay? This is a $1 trillion industry, drug industry. So, you know, when you eat proteins, carbohydrates, fats, your body has carbohydrates, lipases, all these enzymes. They know what are the molecules, what do they look like, they identify them on the basis of topology and then they react, right? But then, what do? You are eating paracetamol or say ibuprofen. These molecules are new, they are made by human beings. How does your body know how to transform these molecules? You eat 5 tablets, then Mr. X will not remain Mr. X. It will stay in your body and then you cannot be your own body. You have to throw it out, right? That is done by the liver enzymes called cytochrome P450. Okay? So, that is called drug metabolism and pharmacokinetics. So, in that, there are hundreds of cytochrome P450. That liver looks reddish brown because of high concentration of those enzymes. So, our research, Danny's and mine, explains how this, see here what I have shown is, these are non-steroidal anti-inflammatory drugs, and then you see the Viagra, Indinavir, then, sildenafil, then ritonavir is an antiviral agent. You see, wide topology, how can the same active site explain? It cannot. So my concept, which involves a different enzyme mechanism, see they all bind far away from the active site. So my theory explains this. It explains drug-drug interactions, how to optimize lead drugs, substrate diversity, so many observations in the drug and metabolism industry. So it has been applied in that industry. Now with a small introduction of what Morborn concept is, with respect to classical enzymology, I am coming to the crux of the talk. What is the crux of the talk? Aerobic respiration. Aerobic respiration is what? If I don't have this procedure, I cannot talk to you, I cannot think, I cannot do anything. All living beings, the most fundamental routine, the way by which you make your chemical energy, that is adenosine triphosphate and heat. Two things that enable you to live, right? You need a powering mechanism. Without powering mechanism, you cannot do anything. Not you, not any living cell. So this is the mechanism that gives you ATP and heat. Now, this is made by my daughter, this cartoon, right? You must have studied in your textbooks, Leninger, Voigt, Harper, Stryer, all that. You might have studied the mitochondria. It's a bilipid. It has two layers, and it has got electron transport chain, chemiosmosis, rotary ATP. For the electron transport, Rudolf Marcus Caltech got the Nobel. Chemiosmosis, Peter Mitchell got the Nobel. For rotary ATP synthesis, Paul Boyer got the Nobel. All right, and then there are so many other people who came afterwards and somehow connected. Many people got Nobel for this. So now, this is the model of aerobic respiration as vouched by the modern day awareness that you see in textbooks. So, I am going to challenge this in a very brief manner, though I would have loved to speak about it in detail. I will rather stick to my theory in a greater extent. If you are interested, I would urge you to ask me questions in the interactive session. Please stay with me. Remember the slide number, or we can just go back and forth later on. So, there's the components are electron transport chain, chemiosmosis, rotary ATP synthesis. I guess all of you would have studied these terms one time or another if you have crossed 12th class, right? Have you studied this term? Yes? I guess so. So, now, these are the protein complexes. Thank you so much. There are so many chairs here. Some of you who are sitting there, you can come here because it's pretty stretching that way. So, now you see, these are complexes 1, 3, 2, 4, and 5. The classical theory says that the complex 1 to 4 are electron transport chain components. Okay? And complex 5 is the ATP synthesis agent. And I'm not going to go into the details of the equation, but there are two different aspects. One is the electron transport chain, and another is the ATP synthesis phenomena, which is connected by chemiosmosis, proton pumps. Now, the mechanism, the analogy is like a car. In the car, what happens is, in the engine, you burn the fuel, and then the pistons go up and down, the expanding gases push the pistons, the up and down motion is converted to a rotary motion. That rotary motion is used for using a dynamo alternator. It is used to recharge the battery. Now, you can say the battery can be, storing the battery can be seen as chemical energy storage, is ATP synthesis. The drives and regulators of the engine can be called as chemiosmosis. What happened? Did I switch off or something? Okay, so now the alternator can be, recycling logic can be called as proton pumps and chemiosmosis. See, the electron transport chain can be equated to the fuel burning engine. Okay, so this is the working logic of mitochondrial oxidative phosphorylation. Now, I am going into critical dissection. This is what each student of science should start doing. If you are taught something, what is the difference between a second standard, third standard child and a person who is doing graduation or post-graduation? When I was taught this subject in my graduation and post-graduation, I never could buy this. Danny will remember in 2009 I said, I don't buy this Peter Mitchell's theory. And then I went on to question it. Only if if it irks your scientific temperament, can you bother to question something? So this I question. See, it's a vitally deterministic scheme. Electrons along, they come like this, go through this. How can it go like that in a mitochondria, which proteins are randomly distributed? There cannot be a circuitry. For circuitry, you need wires connection like that. So I did not. And then oxygen is available everywhere, right? So then chemiosmosis, first you have to pump protons out, and when they come in, how can such a mechanism form at the very beginning? Okay? Then mitochondrial oxidative phosphorylation. You know substrate level phosphorylation? It has lower efficiency per the current theory. Why should evolution opt for oxidative phosphorylation when substrate level phosphorylation is more efficient? Like that. Why do we have reactive oxygen formation, species formation in mitochondria? Life has come so far, it should have minimized it. Another question. Then, why is complex 5 an ATPase? Complex 5, the agent that is supposed to make ATP in our mitochondria, it is actually an ATPase. It has more than 10 million times higher affinity for ATP compared to ADP. If it is supposed to make ATP, it should have higher affinity for ADP. It is natural. But then they saw completely opposite. But they didn't have any other candidate. So they said that this is the ATP. Most importantly, one evolutionary mandate is all those animals or cells that metabolize at a higher rate die quicker. So many millions of years of life, and if metabolism, the energy forming is the most fundamental routine, you would imagine that, the more you do it, it should get optimized. Why should you have metabolism killing your life form? So these are some of the evolution-based questions. The most important argument against chemiosmosis, which even a 12th standard student can make out, is, please see this. This is very important. Protons, you can calculate. Based upon dissociation constant of water, you can calculate. pH is 4, pH is 7, whatever the pH, you can calculate, given a volume, how much protons will be there, free protons. In a mitochondrion, which is only a micron dimension, if you calculate the protons, it comes to 5 or 6 protons in a mitochondrion. It comes only 5 or 6. And can you imagine how many proteins are there? Complex 1, complex 2, complex 3, complex 4? Lakhs. Tens of thousands to lakhs. So, do you think tens of thousands of proteins are present to pump 5 or 6 protons? Impossible. That's the first thing that even a school level kid can do. But this was given Nobel. Peter Mitchell got a Nobel for this. I have published this work. I have taken it to Nobel laureates. Nobody can answer anything. Alright? So, in science you can say what is wrong. You can never say what is right. So, definitely this theory is wrong. Okay? But you are studying this. You will need to study this. You will have to write these answers for quite some time. Okay? Don't worry about that. Okay, now electron transport chain, the other concept. One is chemiosmosis. Even otherwise, what is osmosis? Osmosis is the movement of solvent across a semi-permeable membrane, right? What are protons? Mitchell proposed movement of protons. There is nothing like that. Protons are at the most solutes. They are not solvents. So, in terms of semantics, in terms of thermodynamics, I have shown that thermodynamics fundamental laws Peter Mitchell's theory violates. So, now if you see this, oxygen coverage, oxygen is a diradical. diradical, you know what? There are two unpaired electrons. Now, of course, there are spin barriers and all that, but still, it has got a coverage of 10 to the power 2 angstrom per microsecond. Biological electron transfers are supposed to occur 10 to 20 angstrom distances in microsecond scale. So oxygen will shunt all electron transfers in the ETC. If you see, this is complex 1 structure. Complex 1 structure, electron transfer is supposed to occur here, but proton pump is supposed to occur here. Electron transfer is supposedly occurring in microsecond time scale. Proton pump occurs in millisecond time scale. How can they connect? There is no connection. Just because people believe this is what they have been told, it became like that. So when you question, there is no answer. So ETC is also not a very sensible theory. So oxygen will shunt all that. Then how does, if it is through such a selective affinity based electron exchange, then why should oxygen be toxic to anaerobes? That's a question that the ETC concept doesn't explain. Then in any of these systems, we can put in electron and take out electron through diverse molecules. How is that possible? That is also not explained. Then the third part is ATP synthesis. Okay? The ATP synthesis, it goes round and round. Protons come in. That is supposed to drive ATP synthesis. What are the arguments? Complex 5 affinity is 10 to the power 7 times that of ADP. Then concentration of ATP in physiological mitochondria is millimolar levels, whereas ADP is in hundreds of micromolar levels. So complex 5 will always bind ATP, not ADP. So it cannot ever make ADP, and transmembrane potential is continuous, so that if at all some configuration is changing and then complex 5 is supposed to get that field, okay, now I bind to ADP, I change ADP to ATP, then the transmembrane potential should change, but it is stable. That is also. Then this thing is very important. If you take the equilibrium constant of the reaction, ADP plus phosphate gives ATP plus water, you can use the equilibrium constant to calculate the amount of ATP that is generated. You will only get nanomolar to picomolar level of ATP. You cannot get millimolar. So this theory is absolutely false from a thermodynamic, kinetic and chemical reaction perspective, and structure function correlation also. I will come to that. So now in the last 10 minutes we have, what have I done? I gave you an introduction to Mormon concept. I gave you an introduction to the standing theories on aerobic respiration. Now I am coming to the new perspectives on aerobic respiration, my explanation. Now this might look a little bit overwhelming. Don't worry about that. I will give you a simple explanation. These are the erstwhile electron transport chains components. You see, what my explanation says is, all these complexes have ADP binding sites, which we have shown, and they bind ADP and they use oxygen. All the earlier theory, only complex 4 used oxygen. Now what I am saying is, all these bind ADP, all these make oxygen, all those make ATP. Complex 1 through 4 earlier didn't have role in ATP synthesis. The new theory says complex 1 to 4 makes ATP, and complex 5 doesn't have ATP synthesis role in a direct way. Complex 5 breaks ATP to bring proton in. NADH gives 2 electrons but only 1 proton. So Now, this proton deficiency is what gives the transmembrane potential. Earlier they thought transmembrane potential drives ATP synthesis. It is not. Suppose if a vehicle runs, you get sound and smoke. It is not the sound and smoke that makes the vehicle run. Similarly, in this system, it is the transmembrane potential results because of oxygen's reaction with NADH to generate reactive oxygen species. That's what I have shown here. Okay, I will go to simpler diagram. So, I don't know if you know, this is very subject specific. There are even complex 1, complex 3, complex 4, they are supposed to get together to form respirasomes. Now, how the modern day theory is absolutely a failure to explain. Why? Here, the dynamics of reactive oxygen species, the complex 1 produces ROS, the openings of this complex 1 is towards complex 3. So, the way they come together, these structures can be explained by the new theory. And what does the new theory state? The flavin enzymes, complex 1 and complex 2 activate oxygen to give ROS, and complex 4 and complex 3, they are like turbo agents. You know what turbo agents are? You know turbo machines? If the smoke comes out, that is once again given back to the engine to recycle the non-combusted fuel. So that comes out, and then you use basically the reactive oxygen species to make ATP. How? ROS attacks ADP, creates a radical, that radicals attack phosphate, or ROS attacks phosphate, a radical is made and radical attacks. So rather than a 2-electron reaction that was envisaged earlier, this is actually a 1-electron process, which is why mitochondria has reactive oxygen species. Highest, we'll take your questions in the end. Okay? So this is the explanation by the new perspective. So these are the equations involved. I will not get into the details. Those who are interested in the equations can come to me later. You must remember there was no equation whatsoever in the earlier system for ATP synthesis. It was ADP plus PI gives ATP plus water. This theory is not that. This theory says NADH plus ADP plus phosphate plus oxygen gives NAD plus ATP plus water plus ROS. It's a new equation. It's a completely different equation. If you take this equation and analyze thermodynamically and kinetically, you get all that is explained. Everything beautifully comes out. And believe me, my dear students, this is the most fundamental information you as a biotechnologist or biochemist can ever aspire for. Because on this rests each one of your survival or sustenance or everything. Every molecule, every cell of your being depends on this phenomenon. So while oxygen goes from oxygen to superoxide to peroxide to hydroxyl radical and hydroxide ion to water, through this process, these radicals attack ADP and make water. That's how ATP is formed. Very simple chemical reaction. If they put qualapi, we had complicated it. Why? Because we always thought radicals are dangerous, radicals are chaotic. That's the reason. Okay? So now what are the evidences that I gave, our group gave to support the new theory? The new theory is supported by simple comparison with the cytochrome P450 system. Cytochrome P450 system is a less complicated system. It has also got flavoproteins, it has also got hemoproteins. Now, if you see the consumption of oxygen and the depletion of the reduced nicotinamide adenine dinucleotides, both systems use NADH or NADPH and they use oxygen. Same profile you get, same kind of mechanistic concepts were involved. These kind of proteins are also involved in mitochondria also. So the only thing that common is reactive oxygen species. They use reactive oxygen species. Okay? And then phosphorylation is also seen in mitochondria, albeit at a lower level. So This is transmembrane potential. If you see, at this transmembrane potential only ATP synthesis occurs. Here is ATP synthesis. It starts only at this TMP. Without ROS, there is no ATP synthesis. So ATP synthesis is directly correlated to ROS. And then, the more you give NADH and O2, the more ROS you get. Very simple experimental support. Then complex 1 and 4, independently, each of them can produce ROS, reactive oxygen species. And then increase in ROS, increase in TMP, ATP production is not seen without ROS. Okay? Direct evidence. And the evidence that we gave, you take ADP phosphate in a test tube, you put superoxide into that, you get ATP. Simplest evidence, in vitro evidence. And then some years ago, a lady called Mailer, she had shown in mitochondria that if you put xanthine oxidase, which is known to generate superoxide, you get higher ATP. So that is in vivo, in situ kind of evidence. In chloroplast does photophosphorylation. You might have studied. ATP synthesis also occurs in chloroplast. So now this theory is valid not only for cellular respiration, it is valid for photosynthesis. What sustains life on planet Earth? That's what I'm talking about in terms of trapping sunlight as well as generating chemical and heat energy. So then the other evidence is there are more than 31 electron redox species in mitochondria. The current theory is it is a 2 electron paradigm. Then why should there be so many 1 electron species in mitochondria? Okay? There is no specificity of electron transfer. It's just a wishful thinking that oxygen sits at cytochrome oxidase waiting for 4 electrons and 4 protons to come by to make water. Just to make water. The current theory says oxygen is needed for what? To make water. Nothing else. So it's a transgression of all kinds of rational thought. Oxygen is needed to make water. Why? What is that? You can have water from outside, right? So, now the other things that are involved, the structure and distribution. Now I showed you the structures of the mitochondrial complexes. Sorry, I will go forward, those slides are closer. So these are the mitochondrial complexes, right? These are the mitochondrial complexes. Okay, now you see, these are the transmembrane regions, and there is a large pendulous part subtending down. You see, why should these parts be there? Why should these parts be there? Particularly complex 3, there is no role whatsoever. If no period protein part, it has no role. Why? Evolution doesn't do such things. So, the earlier theory doesn't explain why should there be such pendulous suspensions or the distribution. For every complex 1, there are 3 complex 3s, then 5, 6 complex 4. For the electron transport chain, there should be 1 complex 1. Similarly, it's an electron transport chain. There should be a congruence of the number of components. It is not there. So the current theory says the radical species generation should be very low. Its stabilization agent should be high. So complex 4 and complex 3 will be higher than complex 1. The earlier theory requires it to be 1:1 kind of stoichiometry. So the structure and distribution, now my theory, the new Murborn theory, the proteins could be randomly distributed. It's a stochastic paradigm. The earlier one was a deterministic paradigm. You need to have 1-to-1 connectivity. So these are the structures. So the structures, say coenzyme Q, earlier theory, this was supposed to take, you might have studied CoQ cycle, right? CoQ cycle, The complex 3 is supposed to bind to reduced coenzyme Q, oxidized coenzyme Q, what and all at the same time. In terms of chemical reactions, you cannot have multiple molecules binding at the same time and detaching and all that. It's a highly deterministic scheme. In Tamil, if you say aasa dosa apply vada, that is how it comes to. You might wish for it, it might not happen. The probability of it, chemical reactions are spontaneous collisions. So these are all wishful thinking. If you see study the proteins for the cavities, oxygen access channels, beautifully you can see. This was done by his friend Abhinav. So we showed that they open up towards this structure like that. Then cytochrome C is a protein that is found everywhere. They think that this binds to this and gives electrons. We showed in Danny's work, we showed that it is through a diffusible species. Very clear. There is no specific binding based nothing, because that kinetics is infinitely slower. So, on the basis of, and we showed the same logic in prokaryotic system also, E. coli, same kind of structure function correlations. Very importantly, complex 4, which is supposed to bind to oxygen, we showed that in there is a pseudomonad system where it produces 2 types of complex 4. One at high concentration of oxygen, one at low. At low concentration of oxygen, there was no ADP binding site. At higher concentration of oxygen, there was an ADP binding site. So complex 4, which was supposed to pump protons, all these complex 1, complex 3, complex 4, to pump protons, it is actually ADP binding and ROS generating proteins. That's all. So we have given better function structure correlations. Very importantly, we have shown that heat generation can be better explained. Once again, it's also Danny's work also here. Here we shown that the protons, when they are pumped, this is through an exergonic process. When it comes in, it must be an endergonic process, but then it is supposed to be an exothermic process also. Thermodynamically it doesn't make sense. What we showed is when the ROS react among themselves, it gives heat. When they don't react with ADP and PI, they give heat when they react among themselves. And then the uncoupling protein that is shown here, this uncoupling protein is an anion transporter which transports the negatively charged ROS species outside and that react among themselves to give water and heat. So now the Muralian model of ATP synthesis is rather like a nuclear reactor. It is a stochastic scheme. Now, if the Muralian theory was right, you would imagine a spontaneous nuclear reactor to be formed. There is evidence for a spontaneous nuclear reactor. In Africa, in Gabon, there was a nuclear reactor that functioned for hundreds and thousands of years, spontaneously formed. Water comes into a uranium deposit or cave, then there is deuterium and all that, then the moderation occurs, then there is a critical acquirement of the terms for the nuclear reactor to start functioning. It functions, generates heat, water gets evaporated, then once again water comes in. It worked like that for hundreds and thousands of years. Similarly, you can now correlate the components of the mitochondrial system to similar. See, in nuclear reactor, it is a neutron, right? High energy neutrons split and they go and further divide. In this system, it is reactive oxygen species, electrons. Only thing is here, ROS are the moving things which are being manipulated by the complexes. That's all. Moderated and manipulated. That's all. That's the difference. Now, this is something you can all relate to. I'm coming towards the last part of my talk. Application of the Muralian concept to explain things in aerobic respiration. I was having a discussion. Madam asked how I was talking about cyanide inhibition. Now, currently cyanide is supposed to bind to the ion centers. It is supposed to be, like what madam said, it's a strong field ligand. It's supposed to bind to ion centers and inhibit oxygen utilization or transport. That is the current theory. You might have heard, right? You have a few milligrams of cyanide, the LTT agents and all used to carry cyanide, right? You might have seen so many detective stories and all that. So consumption of 15 milligrams of cyanide in your blood is enough to kill you as a human. LD50 of cyanide is in the range of milligrams per kg. Now, if the erstwhile theory is true, cyanide must bind to iron. If you calculate the blood hemoglobin, roughly you need 10 to the power 22 binding sites, 10 to the power 22 binding sites. Okay? Now, the lethal concentration of cyanide is only 10 to the power 20. So it's 1/100th. Right? How can it be a binding based explanation? My theory is oxygen produces superoxide, superoxide makes ATP, but cyanide ion interferes with superoxide reaction with this. That's the explanation. Now, how do we prove it? Very simple. Not one point of this is my data. This is the best thing. This paper recently got accepted in toxicology, and that is a turning point for our works to get recognized, I believe. Okay? Now this, if you see, carbon monoxide is a much stronger ligand than cyanide. Ordinary smokers have 1 to 2% carbon monoxide. The lethal loads for cyanide, 1 to 2% is already achieved. So, carbon monoxide, even if you take 40%, you might not lose consciousness. 2% cyanide, you are killed. How is it possible when, see, the binding terms of carbon dioxide, carbon monoxide or hydrogen sulfide, toxic gases, they are much better than cyanide. You see, these are the K-on, the rate at which cyanide binds the heme protein cytochrome oxidase. Then Kd is the dissociation constant, which defines the affinity for the ligand with the protein. Then Km or Ki, IC50, inhibition concentration, 50%. These are the mathematical analytical points. The quantitative analysis does not agree even a little bit with the current theory. If the current theory is true, you should have cyanide in breakfast. You will have, no, you have to eat this much, and by lunch you might get killed. See, one simple thing, you see this. The binding of it is in 10 to the power 2 per molar per second. The cyanide concentration that is lethal is micromolar. Multiply micromolar into 10 to the power 2. What do you get? 10 to the power minus 4 per second. It will take hours. But if you went back, you saw those radical reactions that I told you were the supporting theory. It's not going back. Yeah, this one. These have these radical reactions 10 to the power 10, 10 to the power 10, very fast. So these can explain the outcomes. So only the Ross mediated reactions explain the outcomes quantitatively. So cyanide, even otherwise, Km of oxygen is much lower than Kd. How can it be? Km, if you study basic Michaelis-Menten kinetics, you know Km is equal to Kd plus K2 by K1. So Km can only be greater than Kd. It cannot be less than Kd. So that is something not seen here. So the whole conceptual grounding of this theory has been shown to be redundant. So next is the theory. So what does happen is hydrogen cyanide diffuses into the mitochondria, reacts with hydroxyl radical, giving the cyanide radical. Cyanide radical reacts with superoxide, giving cyanide ion. This hydroxyl radical and superoxide radical have been rendered water. So, the radicals that were supposed to make ATP, they are getting converted to water without getting any functional activity. That is the cyanide's role. That's what we have shown here. So, erstwhile, complex 4 was supposed to just take 4 electrons from cytochrome C, pump protons and make water. This is a not possible reaction. That's what we say. So, the second one is the powering logic, homeostasis. Why is water needed? What does proton do? This is the cellular cycle. The earlier cycle had closed mitochondrion in which protons were pumped across the internal membrane to the intermembrane space and the proton was cycled there. This is a non-working logic. There is no logic for that. Okay, we have shown that. Here the protons moving from intermembrane space back to the matrix is what gives heat. So this is a closed logic. There is no way in which you can have the NADH, it gives only one proton but has 2 electrons. It doesn't give how that proton gets supplemented. Now here is this reaction. The new model, it explains homeostasis. Homeostasis na, suppose an RBC has 1 sodium inside whereas 20 sodium outside, 40 potassium inside, 1 potassium outside. How? This is the mandate of homeostasis. Simple, I am not talking about that here. Homeostasis is how a cell maintains its component. Now, if you see the equation there, the equation that I have shown for Murban concept, ADP plus PI plus NADH plus O2 gives ATP plus water. So what is happening is, if you see the colligative properties, the number of solutes that are formed are lower than the number of reactants. So the water that is formed inside will go out. You know, that is real osmosis. This is a physical phenomenon that has been established. There is no doubts whatsoever. Osmosis nadakko. So water that is formed here goes out. So protons from outside come in and water goes out. Fuel is present, injected, goes out as smoke. You have a working machine logic. Same working logic, adhe pola irukra working logic. So for the first time, we have an explanation of how a cell functions with respect to the fundamental powering logic. So I am coming to the end of my talk. My talk was prepared for 1 hour. Correctly, I am coming to that. So basically, summating your textbook awareness of what life is about, how ATP is generated. Currently, NADH or succinate gives electrons to the electron transport chain, 1, 3, 4, 2, all complexes. They are supposed to relay through a circuit, and while the electrons are going through them, protons are pumped by the electron transport chain into the intermembrane complex. This gives protons more outside, giving positive charge outside in the intermembrane space, and thereby a transmembrane potential is generated by the osmosis, chemiosmosis process, and then this proton motive force, while it is coming through the complex 5, the complex 5 goes round and round making ATP. This is the current theory. Okay, so there are 2 equations, independent equations. You see the comparison. Now, on the contrary, the Murban scheme is very simple. Complex 1 and 4 are membrane proteins that basically bind ADP, and they also activate oxygen. Complex 1 and 2 activate oxygen. Others use different reduced coenzyme Q or cytochrome C. Cytochrome C is present to gather lost electrons. So that's why when mitochondria ruptures and cytochrome C is released, you have apoptosis. Evolution has learned that when the mitochondria ruptures, it's better to kill itself. Reactive oxygen species are going to be flooding plenty, and it will lead to a lot of problems. So they find one simple equation defines ATP synthesis. NADH plus oxygen plus ADP plus PI gives NAD plus ROS plus ATP plus water. ROS neutralization is what gives heat. So all roles are redefined, completely different explanation. There is no commonality whatsoever from your textbook explanations. So this is the new explanation, and we are at a very interesting juncture now. I started working on Morborn concept in my late 20s or early 30s, and I had a tough time trying to convince people. If you are embarking on anything important or interesting in life, you might not get people supporting you, appreciating you at the very outset. Today I am not a recognized person, but I know what I am doing is right. Because, why? Because mathematics, physics, fundamental laws are with me. You know that. So this is a conference on transdisciplinary subjects. So please get your fundamentals in maths, physics, chemistry properly grounded. And with this I can take on anybody. That gives me the confidence. I was also a biologist like you, but I took the time out to learn my fundas properly. So I know I'm right. I'm not going to be like this guy. I will not give up. You will hear about me. I'm challenging you. I'm telling you. So I'm not going to give up. I'm there. I will continue. It's a pursuit of 20 years. So it's a paradigm shift kind of work. The paradigm will change. Maybe your kids might get to study a different theory in their textbook. All right? So it takes time. You know, Thomas Kuhn has studied paradigm shifts in science. So there's a new idea like mine doesn't become a fact in science. It takes significant amount of time. You know who's Max Planck? Quantum physics, right? E is equal to h nu, that person. He said a new scientific truth does not triumph by convincing its opponents, but rather because of a new generation grows up that is familiar with it. You are the new generation. You are the new generation. You are going to be familiarizing with this, and you will usher in the new paradigm. Do you understand what I'm saying here? This is very important. And scientific pursuit depends on falsification. It's very important. Very clearly, we have shown that the current paradigm is wrong. Our theory can also be wrong. But then, until when it becomes wrong, we have proposed the standing theory. That's what I'm claiming. I'm not saying my theory is correct. I'm just saying my theory appeals to what is known till now. Okay? So now in terms of, in terms of further applications, this is going to explain photosynthesis, epigenetics. How do genetic level, two twins will have the same DNA. How do you explain the difference? By Ross chemistry, DNA acetylation, modulation, all that. Epigenetics, it is going to be cybernetics. How do you have memories? How do you have memories? It's all by Morborn concept. All things, everything is going to be changed. This is the most fundamental discovery in history of mankind. I boast because of course I'm the discoverer, so I will have to say that. But then it is, because it is going to change practically everything that we do hold as true. So you are seeing me. What you see is an image on your retina, right? Now this image has to be processed to an electrical signal. It has to go to your brain. How does it happen? Current theory says opsin, conformation change. So how does a conformation change translate into an electrical signal? No answer. This gives an electrical signal. Radical. Involvement of radicals have been overlooked in biology for the simple reason that it is supposed to be chaotic. But then I showed you chaos has inherent order. From chaos came life, to chaos goes back life. So, as young students, you should always remember to explore. I told you the second philosophy, explore, resolve, prioritize. Chance favors the prepared mind. Explore, resolve, prioritize, pursue excellence based on what you have and what you know. This exploration has been highly overlooked in modern day science. Why? Because everything is based on hypothesis driven science. Before you know what is right or what is wrong, you have already formed a hypothesis. If I ask you, Danny, have you stopped beating your mother? I mean, that question doesn't have an answer, right? Because that question is framed in such a way that it cannot be answered. Many of the scientific problems that we face today are like that. You form wrong hypothesis and you say this can be either a null hypothesis or the hypothesis is ratified. It's sometimes not like that. So we have a problem in modern day scientific approaches. Thank you so much for appreciating. These are my publications in the field. The publications, these are in drug metabolism. It is published in the leading pharmacology journal, Frontiers in Pharmacology. Dose response, that was it's Danny's work. We published in dose response, once again the leading journal in the world. Then debunking chemiosmosis was published. These are published also in journals which have 50, 70 years of tradition. They are decent impact factors and all that. So that's it. And the publications have been given cover page credits in annual journals also. So it's not something very wrong or misplaced that I'm talking to you about. You can go and verify your textbooks. You can see the conventional perspective is, you know, mitochondria produces ROS and then that gives to diseases, right? You see here. This, but now ROS generation is important for heat, for energy, and then there are certain places where it has problems. You might use in the kitchen a knife. If you don't use it properly, you will cut yourself. Reactive oxygen species have the same relevance. There are cutting boards in the kitchen, there are gloves in the kitchen. Your body has similar techniques. Do you understand what I'm saying? Just because the knife cuts you, you don't stop using the knife. Similarly, reactive oxygen species are used by our body to do the most important functions in our body. That's the reason why we need oxygen. That's the reason how cyanide kills so quickly. And thank you so much for your patient attention, gathering. And now I hope at least a few of you will ask questions. The majority of you have been, I think, have been involved in your own discussions there and there. But there are a few people who have paid attention, and I hope that I am useful to them in the next 15 minutes or so.